Chapter 4, the tissue level of organization. There are 4 types of tissues that we're going to cover in this lecture. The first being epithelial, the second being connective, the third being muscular, and the fourth being nervous. The first tissue we're going to look at is epithelial tissue. So let's look at some characteristics of this tissue type. Epithelium forms continuous sheets of cells. There is little to no intercellular material between the cells, which means they are tightly packed. Cells are going to be attached to a basement membrane. It is avascular. Now remember, avascular means that there is no blood supply. Therefore, it's dependent on diffusion of nutrients from the connective tissue layer below it. It covers surfaces, the external body surface, and forms a protective barrier that regulates permeability. So if you think about our skin, the largest organ, what you see on top is our epithelial lining. When our surface cells die, they are replaced by the division of cells below it. Epithelia can also be organized into glands. Functions of epithelium. Epithelial functions as a physical protection and maintain integrity. If again you think about our skin is here with modifications of keratin, which hardens and deadens that upper cell layer, prevents dehydration. It also protects us against mechanical or chemical abrasion. If you think about mechanical abrasion as if you bump into a table or scratch your skin, we're not down into that vascular layer bleeding out every time. Chemical abrasion, same thing. Rain, if a bird were to poo on you, or if somebody were to spit on you, all those enzymes wouldn't break down and get into those lower layers. Epithelium also serves as a function for transport, regulation of nutrient absorption and ion transport. So again, if we think about the digestive system, we know that the function of the digestive system is to transport nutrients, right? So absorption of nutrients. And in our kidneys, we're regulating ion transport and absorption and secretion of ion. So this epithelium is going to set up these routes. The last function is going to be to provide sensation. So we have these large sensory nerves that are going to detect stimuli, things like temperature, hot, cold, pain, pressure, light pressure, tickle, itch, some deep constant pressure. And again, this will give us feedback to the central nervous system to respond accordingly. So we're providing, again, sensation. Let's look at the organization of epithelial cells and their basement membrane. So what we have here is our epithelium, right? And we're going to classify epithelium based on shape and arrangement. So right now, don't worry too much about the shape, but just look at the cells. We have a couple landmarks. The top is called the apical or free surface. Where the cells touch on the side are known as the lateral surfaces. Where the cells touch the bottommost layer is known as the basal surface or bottom. From there we have the basement membrane. This is what's connecting this cell layer to the connective tissue layer below it. So we have first our basal lamina, followed by our reticular lamina. When we look at the composition of these lamina, the basal lamina is made up of collagen fibers, okay? And the reticular lamina is formed by reticular connective tissue fibers. And when we do connective tissue, we'll look at this a little more in depth. Classification of epithelial tissue. So we're going to look at how we classify epithelium based on shape of the cells and arrangement of the layers. Now, we can do this via a very simple way. We can start with this 2-word nomenclature. Now remember, when we talk about epithelium, we are always using a 2-word nomenclature based on number 1, arrangement, so this is what we start with, and number 2, cell shape. This is going to be our predominant method throughout the entire nomenclature of epithelium, except for one type that we'll introduce at the end of this tissue lecture. So let's look at our arrangement. This is pretty straightforward. We can start with looking at the basement membrane. We notice and count how many cell layers move upward. So we see that this picture shows one box next to another, next to another, next to another. So when we look from the basement membrane up, it is only one cell layer thick. We will call this simple. We're going to go over here to the end. Again, we start at the basement membrane, so we're working from the basement membrane to the apical surface, so to the top, and we count the cell layers. 1, 2, 3, 4, or more. So any cell line that has 2 or more cells thick, right? So we go more than just simple. So 2 or more, we're going to call stratified. Now we have this outlier, and we can break apart the word pseudostratified. Pseudo means fake, stratified means layer. Now again, we look at the basement membrane, and we get these really tall, elongated cells. But we notice that it gives the impression of being stratified. But if you look at the basement membrane, they all line up 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, etc. The problem is that these cells, think of it as they're getting lazy. They're so tall, they start folding over each other. And they give the impression of being stratified when in actuality they are not. So we use the word pseudostratified. Next is going to be based on cell shape. We have 3 options here. So there's no need in reinventing the wheel. We keep it pretty simple. 3 options for arrangement and 3 options for cell shape. So again, we're always looking at the basement membrane when we're talking about epithelium, and we see that this cell is flattened, almost like a fried egg, okay? 1 cell layer thick, flattened. We're going to name the cell shape. We're going to call it squamous, okay? Look at the next one. Like a perfect box. We're going to call it a cube or cuboidal. Our third option is these elongated column-like cells. We're going to call it columnar. Now, how do we put it together? It's pretty straightforward. We could just simply drop lines to explain the arrangement and shape. For example, simple squamous, simple cuboidal, simple columnar. And we can do the same with stratified. Stratified squamous, stratified cuboidal, stratified columnar. Now with pseudostratified, we're only going to use one type, which is pseudostratified columnar. So we'll draw a red line to explain that pseudostratified columnar epithelium. So don't worry about all the lines. We're going to go ahead and erase them for you because we are going to outline where we find all these epithelium and exactly what they do. Remember that epithelial tissue covers and lines tubes or open spaces and open cavities of the body. For example, we look at the alimentary canal or the digestive canal. The lining of that tube is epithelium, and it is open to the outside air or space. Same thing with the covering, like our skin, open to the outside space. Now we could talk about simple or stratified or pseudostratified. Remember, the name of the specific type of stratified epithelium tissue depends on the shape of the apical cells. So we look at the specific shape. So again, aligning, arrangement, and shape. Table 4.1 in our textbook is going to show us examples of each of the epithelial tissues. Look for the similarities and differences. My suggestion here is to draw and color. They sell coloring books that assist with epithelial tissue. You can watch tutorial videos, my tutorial videos on YouTube to get through some of the differences between the histology. But again, drawing, identifying, and labeling seems to be a good route to help you learn and master these tissues. Let's start with our first combination. We're going to look at simple squamous epithelium. So simple squamous epithelium. So from the terminology, we know its shape and arrangement. Simple being one cell layer thick, squamous being flattened. So let's look at our drawing. We follow the basement membrane. We see one flattened cell, and it is found in various places. So one flattened cell layer, simple squamous epithelium. Okay. When you look at it, it looks like from the apical surface and look down, it looks like a bunch of fried eggs with the yolks in the center. But in lab, we're not going to look at this view. We're going to look at this sectional view to see the side of it. So it will look like this flattened cell type. Now, location. We find it lining the cardiovascular lymphatic system, heart, blood vessels, lymphatic vessel linings, where it's called the endothelium. It's going to form the epithelial layer of serous membranes, like the peritoneum, the pleura, and the pericardium that we introduced in our first lecture. It's called the mesothelium. We also find it in our air sacs of our lungs, and more importantly, the glomerulus or Bowman's capsule of the kidneys. Now, why did I say more importantly the kidney? When you look at this in lab, in a physical lab, or through my tutorial, you're going to find that we do not use the lungs, but we use something called Bowman's capsule inside the kidney. So the kidney is used actually twice, for simple cuboidal and simple squamous. So we look for that renal corpuscle. It is essentially a ball within a ball. The inner ball is a fenestrated capillary, okay, where it's going to be filtering blood. And this outside layer is going to be made up of simple squamous epithelium. So you look for this ball in a ball. And again, you have all these little nuclei forming that wall. So that layer right there is that simple squamous epithelium. Again, look to my tutorial on YouTube to explain this in more detail and see a visualization of this. Now the next tissue we're looking at is simple cuboidal. So again, let's look at the nomenclature. We are looking for one cell layer thick of cuboidal shaped cells. Now, there are trigger words as we work through epithelium and through connective tissue. Here we're looking for tubes. So remember before with simple squamous we said a ball in a ball for the kidneys? Well, here we're tubes, tubes, tubes. So let's look at our tube. We have the lumen. So again, we're in the kidney. So this is where urine is going to be made and moving. And then we have this outside layer that forms the tube. Notice each cell is a block, 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 like a nice cube-shaped arrangement, nice big fat round robust nucleus. So if we move up here, nice cubed big round nucleus, nice cubed, nice round nucleus. It is one cell layer thick. All the area around, all this area is stroma, are other tubules. So look, here's another tubule, right? Simple squamous epithelium, another tubule with simple squamous epithelium. So all these tubes are made up of simple squamous epithelium, one cell layer thick and square. Now let's look at some of their functions. The function of this epithelium, specifically here in the kidney, is going to be for secretion and absorption. As far as location, we're going to pick pretty much one spot, one place to find this, and that's going to be the kidneys. You can go through your book and look at the surface of the ovaries. We can go to the posterior surface of the retina. We can go to some smaller ducts and glands as being simple cuboidal, maybe some glands in the pancreas as well. But again, try to make your studies a little bit easier and focus on one place that we find it, specifically here, as we show in the tutorial video, the kidneys. The next tissue we're going to talk about is simple columnar epithelium. Now, let's look at the pictures, walk and talk our way through it. Find the basement membrane, look at our cells, 1, 2, 3, 4, 5, 6. They're lined up like soldiers. This is one cell layer thick, they're elongated, so we use the word simple columnar epithelium. Now, where are we going to find this? We're going to find this in the stomach, okay? As well as the small intestine and large intestine. We're going to see ducts and glands and the gallbladder. Now, why is this important? Well, we have to now talk about modifications. So we're going to drop ourselves a nice little box covering all this stuff, and we're going to write down our modifications. So modifications are found typically within or on the surface of epithelium. What do these modifications do? They're extensions of the cytoplasm that serve specific functions. Our first modification is goblet cells. Now, we say, what do goblet cells do? Well, they're going to secrete mucus onto the surface of this tissue. So we look back to our drawing, and here these little bulbous goblet cell shaped structures, right? And we look at the drawing, here it is. This is going to secrete mucus onto the surface to help facilitate movement, to lubricate, and also for some absorption. The second modification, especially one that we see here, is microvilli. So we say, well, what's microvilli for? So think about the small intestine. What are we doing there? We're absorbing nutrients. So if we're absorbing nutrients, what's the next thought process? We need to maximize surface area. So these little finger-like projections are there to maximize surface area. Why are we maximizing surface area? Absorption, maximize absorption. And we see that here. Look closely on the surface. We get this brush border, and you can see it here too, right here on that apical most surface. That brush border are little million finger-like projections on top of already finger-like projections. So villi with microvilli. So again, maximizing surface area. The third modification we're going to see is on our next slide, and it is cilia. So hair-like projections that are going to help to filter, broom, catch, and move stuff throughout the body. The fourth modification we're going to see on the skin is keratin. And this is a protein modification that's going to harden, deaden, and waterproof our skin. Now that we have the four modifications, keep these in mind in your notepad or in your brain as we continue through this lecture. The next tissue is going to be simple columnar epithelium with cilia, so ciliated. Now, let's look. One cell layer thick. Look, they're lined up like soldiers. We find these in the fallopian tubes. We find the apical surface. All these hair-like structures. Look how much bigger and broader and spaced out it is compared to our microvilli. Okay, so we get these simple columnar epithelium. There's that basement membrane, right? So one cell layer thick, tall, columnar. We do see some goblet cells and again cilia. This slide is going to show us pseudostratified columnar epithelium. So again, look, elongated, but then you have another one here. It gives this false impression of it being stratified when in actuality it's not. So pseudostratified columnar epithelium. Now what do we see? Basement membrane, apical surface, and all of a sudden, hairs again. Cilia, and some goblet cells thrown in between. So we utilize two modifications here. Where are we finding this? We're going to find this, for our purposes, in the trachea. Now, what's the job? We're building support. We're going to make this tube rigid with this cartilage, this tracheal cartilage that's not seen here, but we will look at it when we do some cartilage. But this epithelium is going to serve a very important function. Its function is to act like, essentially, flypaper. The goblet cell is going to secrete mucus onto the surface. The cilia is going to help to transport that dirty mucus out of the body. So what's happening here? You're brushing or brooming your garage in the springtime or fall time, getting rid of some leaves or dust and dirt, and you breathe in all that dirty air. The goblet cells are secreted. It's going to trap and attach, like glue paper, all that dirt and debris that you're breathing in. The cilia is going to power stroke it, move it out so that you can cough it up and spit it out so it doesn't make its way down into your delicate respiratory membranes. So a very important function. So again, look at shape, arrangement, modifications as it correlates to function. That brings us to stratified squamous epithelium. Now what do we hear in our words? Stratified, more than one layer. Squamous, flattened. So let's take a look. 1 cell, 2 cell, 3 cell, 4 cell, 5 cell, 6 cell, 7 cell. Well, it's not cuboidal. It is not columnar. It is definitely flattened. It's more than one cell layer, looking from the basal membrane all the way to the top. Now that we have identified the shape and arrangement, the next is to look at if there are any modifications, and where will we find them, and where do we find this epithelium. So first, notice that this is non-keratinized. Look at that apical surface. There is nothing there. It ends with the cell line. Right? So it ends with that stratified squamous. Nothing above it. No wispy layer, no protein modification. So this is non-keratinized stratified squamous epithelium. Where do we find this? Well, you'll find it lining the mouth, esophagus, parts of the epiglottis, pharynx, and the vagina. Again, its function is to protect against abrasion. That's why we have so many layers. It also prevents from dehydration or water loss, and prevents or protects against UV radiation and foreign invasion of bacteria. Now we're going to look at the same epithelium as the previous slide, stratified squamous, but now we're going to add a modification. So this is keratinized stratified squamous epithelium. So look at the cell line, flattened cells, multiple layers high from basement membrane to the apical surface. Now let's add a protein modification. This wispy layer here is that hardened, deadened, keratinized layer that's going to line our skin to protect us against abrasion, water loss, UV radiation, and foreign invasion of bacteria, microbes, etc. Stratified cuboidal epithelium. Now, while we don't technically have this in our lecture, or we don't do this in lab, we are going to introduce this in lecture so we understand that, one, it does exist, so stratified cuboidal does exist, and look at the picture. It is cube on top of cube. We'll find that this is organized in ducts of sweat glands and parts of glands in the esophagus, as well as parts of the male urethra. So how do we know? Lumen, it looks like our tubes, but the only difference is we have a box on top of a box, 2 layers high. So because we're 2 or more, we are stratified cuboidal. Its function is protection. We're limiting secretion and absorption that's going on within these tubes. The next epithelium we're going to cover in lecture, but again, you won't be responsible to know in lab, is stratified columnar. Again, we need to know it exists. Elongated cells stacked on top of each other, so stratified columnar. But again, you will not be responsible to know this in lab. What's its location? Parts of the urethra, large excretory ducts of some glands, such as the esophageal glands. We find some areas in the anal mucous membrane and parts of the conjunctiva of the eye. Its function is protection and secretion. Now let's look at the apical most surface. Do we see any modifications? The answer is no, not here. Our last epithelium is transitional epithelium. So notice that we don't have a 2-word nomenclature here. This is that one time that I said that we will not have to use 2 words. We could use one, transitional. Let's look at its name. What does transitional mean? Well, transitional means changing or transitioning. So these cells change shape from more squamous and cuboidal looking cells to columnar and cuboidal looking cells through the surface. So where will we see a changing in shape based on a changing in organ? Well, the answer is right here, in the urinary bladder. So when the bladder's full, these cells would look more flattened and cuboidal. When the bladder is relaxed, the cells will take on a more cuboidal and slow columnar appearance. Okay? Now, let's look at this picture. Pretty high. So we say that the bladder is empty here because we see that the cells are relaxed. They're not squished down. They're pretty big and robust and rounded. So look how round these are. Okay? Then we go to the slide. And we say, they look more flattened with cubes. So what's going on here? This is the bladder when it's filled. Look how much thinner that epithelial lining is. Let's summarize all the epithelial tissue and possible naming combinations, especially what you're going to see in lab. Okay? So, like we did on our first slide drawing our lines, you will be required to know simple squamous, its location, identify it, simple cuboidal, its location, and identify it, simple columnar, its location, and identify it, pseudostratified columnar, its location, and be able to identify it histologically, stratified squamous, keratinized, and non-keratinized. You need to know for lecture that stratified cuboidal exists, but you will not do this in lab. Same for stratified columnar. And you need to know for lab transitional. Do not forget to include your modifications. When you're identifying epithelium, these are your cheats. This is what's going to help you identify them. You see cilia. We only do one slide that has cilia, and that is pseudostratified columnar, ciliated. We do microvilli. We only do one slide that has microvilli. Simple columnar is the only one with microvilli. So use those cheats to help you identify the histological structures of this epithelium. Let's look at some clinical connections between epithelium and how we can use this in a doctor setting or clinical setting to set or diagnose different conditions or pathologies. So we're going to look at something called exfoliative cytology. This is the study of cells removed from epithelial surfaces. Why would we use this? We use this to examine for conditions like cancer of the skin, respiratory cancer with something called a fine needle biopsy, or digestive system diseases. We can also do something called an amniocentesis, which is a fetal testing, which is collecting cells from the amniotic fluid, which is used to test for trisomy 21, which is also known as, so trisomy 21, which is also known as Down syndrome. You also get this done when you go to your OBGYN yearly for pap smear testing. So we're testing the cells of the cervix, looking for cervical cancer. So very important that we understand epithelial anatomy and physiology so that we can understand how they are affected when exposed to disease or disorders. Glandular epithelium. So a gland is a single cell or a mass of epithelial cells adapted for secretion. We can classify glands via two mechanisms. Either they are endocrine or exocrine. Let's look at our terminology. Endo, meaning within, and exo, meaning outside. So we think about endocrine. Endocrine glands are going to distribute or push stuff, hormones, but for now we'll call it stuff, inside or within the body, more specifically the blood. Whereas exocrine glands are going to push, again, stuff, we'll elaborate and be more specific in a little bit, but push the stuff to the outside of the body or on surfaces like our skin or on the surface of the tubes of our digestive tract, etc. Let's look at a sectional view of a secretory portion of an exocrine gland. So this is an eccrine sweat gland. So if you think about sweat, its location, we're talking about in the dermis or the skin region. We're talking about pushing water or waste out onto the surface. So this is an exocrine sweat gland. We can look at the epithelium and classify the epithelium as well, right? So 2 box high, there's that basement membrane. Here's that apical surface that sweat's going to be pushed out to the surface. So this is a stratified cuboidal epithelium. So a nice eccrine sweat gland. We can now look at the structural classification of glandular epithelium. They can be, as we said before, single cell or unicellular. They can be comprised of many cells called multicellular, and the ones that are composed of many cells, they form a distinctive microscopic structure or macroscopic organ. For example, we have our eccrine sweat glands, we have our sebaceous oil glands, so we can have eccrine, and then we have sebaceous, and then we have our salivary glands like our parotid gland that's going to secrete digestive enzymes. So we'll elaborate more in a little bit. Let's look at some examples of different types of glandular epithelium. The first one we're going to look at is called a merocrine gland. Okay, so we look at our picture and we'll look at the terminology. So the products are released through exocytosis. So remember in our previous slide, exocytosis is taking a vesicle, fusing it to the cell membrane and pushing it out its surface. And then we have an example. So we find the example of mucin secretion in the respiratory system and the digestive system. So we're talking about mucus. In skin, they produce watery perspiration. So let's look. Let's look at all of our cell components. We have the nucleus. We have the rough ER that's making this product. The Golgi apparatus is packaging it into nice, neat little vesicles, and then it's being exocitized out. So pretty productive way to make, in this example, some saliva. The next gland is an apocrine gland. So apo means top. This involves the loss of cytoplasm and the secretory product within. An example is milk production, the mammary glands. So look what happens. We essentially pop the top. Similar process, the nucleus tells the cell what to do. The rough ER is making this milk that's being put into these nice, neat little vesicles. And instead of exocitizing, we're pinching off, breaking off the entire port, secreting this out through, in this example, the nipple. The next gland is a holocrine gland. So let's look at this word holo. I like to think of it like halo, like an angel, right? So this is where the entire cell completely gets destroyed. So the cell is going to become packed with secretory product. Then the entire cell is going to burst. This is going to destroy the gland cell. And then we're going to have to wait for mitosis to occur in order to make more. Where do we see this? Sebaceous glands or oily secretions, which is why when you wash your hair and you strip all the oil out of that follicle, it takes time to make and re-oil that hair. So look at our process. We have a cell that's mature, filled with all of our oil. It pops and destroys, secreting all of its product. Then look below here. We have mitosis. Here's our end stage telophase. So we're cleaving, forming this cleavage furrow, telophase. That's going to make 2 new secretory cells. So again, just to review, merocrine, exocytosis, apocrine, pop the top, holocrine, completely bursts and dies. Let's go back to epithelium. How do we get these layers to stick together, from top to bottom, side to side, as well as how we get them to communicate with each other? So epithelial cells attach to a so-called basement membrane via adhesion molecules called CAMs, so cell adhesion molecules. These are transmembrane proteins that are going to act as anchors to hold the cell in place. Cells are also going to be attached by specialized cell junctions. Tight junctions, which link the lipid portion, so the phospholipid bilayer of the cell membrane, and then desmosomes. And we're going to deal with 2 types, desmosomes and hemidesmosomes. Okay? And this is a little different. This is linking the cytoskeleton of each cell. So let's look at this and think about this logically and put an analogy to it. Tight junctions work if I were to say to you, take 2 people and link you together as strong as you can. You can stick your skin together. While it's a good connection, it's not the strongest. So you're linking your skin, which essentially be synonymous to linking the lipid portion of these cells. Now, if I said you make an even stronger connection, hypothetically, let's say we can fuse your skeleton together. So you cross arms, you link your skin, and now you fuse your skeleton together, side by side. These are desmosomes. But now look, you made a strong connection, but you're not tied directly and anchored into the basement membrane. So now let's take your feet and fuse your skeleton into the basement membrane. This is a hemidesmosome. The next is, how do these cells communicate with each other? Well, those are these gap junctions. Gap junctions are going to allow for cations to move from cell to cell. So they are little pores or channel proteins that act as pores in the walls of the cells that allow for them to communicate with each other. Let's put it to diagram. Here's our tight junctions. As you can see, we have the lipid portion of our cell sticking together, forming these spaces, intercellular spaces, and plaque bands. Another junction is called an adherens junction. So we look at the face of the cytoplasm. So the cytoplasmic face is linked to the actin cytoskeleton. So we have this actin cytoskeleton, this band that is linking together to the band adjacent to it. So it's adhering to another cell wall, linking this cytoskeleton together, similar to tight junctions that regulate passage of ions and small molecules between cells and establish cell polarity. Desmosome. So again, we're linking side by side the cell wall. It's defined as a structure from which two adjacent cells are attached. It's formed by protein plaque, so we can see the protein plaque here, in the cell membranes, linked by filaments. So we have these intermediate keratin filaments. So again, we're interlocking the cytoskeleton of each neighboring cell. So again, like our example where we had two students fusing their skeleton, this is what a desmosome does. Now a hemidesmosome is similar to a desmosome, but instead of locking the arms of a person side by side in our example, it's locking your feet into the floor. So what do these hemidesmosomes do? Same plaquey band, locking with our intermediate keratin filaments, our cytoskeleton, that's anchoring and forming a tough bond with the basement membrane. Remember how we asked, how do cells communicate? Well, here's a diagram. These are our gap junctions. These are pores in the cell membrane that's going to let cations move through the cell, allowing them to communicate. Let's put it all together. So let's circle right here. We have some nice gap junctions. See the little pores that are letting cations to move through. We have tight junctions up here binding that lipid portion of the cell. We have some hemidesmosomes anchoring the bottom of the cell into the wall. And we have some desmosomes right here that's binding the side-to-side wall of these structures together, again anchoring laterally, horizontally, and forming this nice tight anchor to the cytoskeleton.